All right, good afternoon. Let's get started. So just a reminder, we have next Monday off, right? MLK Day, so I won't be here Monday. And you don't need to be either. Also, I forgot to say it last time, but I am recording the lectures. I said that in my syllabus. I'm hopeful that will be helpful for the number of people that are sick right now. I do think it's better that you are in class. So it's good for me to see so many people here. I think you're just gonna get a little bit more from it when you're actually in class, you're collaborating with other people. You can ask questions. So I think that's the best way, if at all possible. I'm not getting as many emails about textbook purchase. So there's still about 30 of you that need to sign up for that. So if you haven't done it yet, remember you need to do that through Save, or I think it's going to be about $10 cheaper if you go through the bookstore's Save program. So check out the link that I have on Blackboard for that. Those of you that registered the way I had originally asked you to through Pearson, that's still fine. Like you can still leave it that way. I have about 30 of you registered that way, and that's fine. If you want to get your money back from Pearson, you would have to contact them and then do it through Save. Again, you save like $10, but it's fine the way it is. If you did temporary access, then you might want to, when you actually pay for it, go through the Save program. If you did register the way I suggested that you do through Pearson, make sure you opt out of the Save program. So I'm not sure how you go about doing that. You might have to contact the bookstore. I can try to find out. Otherwise, they will charge your tuition bill for it if you don't opt out. So I'll be welcome to try to field any questions you have about that. You might be better off like talking to each other to figure that out. But certainly if you have problems, bring that to my attention. And sorry, that was a little, I'm learning too. Okay. So where we left off last time, we just started into chapter 6 and talking about culturing bacteria. Those of you that are in lab got a chance to do that today already. We talked about aseptic technique in lab today. So that's where we ended last time after going through kind of some just, hey, this is what we're going to study this semester, and a little historical perspective. We jumped right into how do you grow bacteria in the lab, a little terminology there. So we'll finish with that today and then move on into microscopy. But where we left off last time, I was talking about how you keep your cultures pure. So remember, a pure culture is one where there's only one kind of bacteria growing. We try to get these isolated colonies. Each of those discrete packets of cells that we call a colony, we assume grew up from a single cell. All right, I don't think I mentioned that last time. So each one of these colonies, they're essentially clones of each other, because the way bacteria divide, they get a little bit bigger and then they split into two. So each daughter cell is the same as its parent cell. All right, so each one of those colonies, it's all the same kind of cells. And when all those colonies look alike, then we think, yep, it's probably everything in that culture is the same thing. It's pure. Okay, and we try to keep that purity by aseptic technique. So that encompasses a lot of things like flaming your loop, like wiping your bench down with disinfectant, washing your hands before you work with bacteria, keeping the lids closed. Okay, those are just some examples of aseptic technique. And again, if you're in lab, you'll learn a lot more about that. All right, so let's talk a little bit more about culture medium. we have a lot of different kinds of media that we use to grow bacteria based on the kind of bacteria and based on what we're trying to grow. So some labels that we slap on media to help us kind of understand what the media can do for us. All microbiological media is either considered defined or complex. Okay, if it's defined, that just means that we know exactly what's in it and exact amounts. So the exact chemical composition is known. Okay, we can write out the chemical formula for all the ingredients in that media. Okay, so let me show you an example of that, and I'll come back to that slide. All right, so this was, I think this was the table right from your book, table 6.3. So this is again, a defined media. Sometimes we refer to that as a synthetic media. And if you look at the ingredients here, you know, glucose, we know the chemical formula for glucose, C6H12O6, all right? We know the chemical formulas there. And I couldn't even tell you like why all of those things are included there, but we know exactly what's there. We don't very often use defined media because it's just needlessly complicated to prepare. Okay, you would have to, you know, find all these chemicals and put them in there. You might want to use this if, say, you're trying to grow only bacteria that can fix their own nitrogen. So you might want to make sure you give them a media that doesn't have any nitrogen in a usable form already. Or maybe you're trying to figure out exactly, you know, what is that ingredient that maybe would an organism would need in order to grow. And so maybe you want to leave, you know, one of those ingredients out. Okay, so commonly, these are not as commonly used defined medias. More often, we're going to use complex medias, and that just means that the exact chemical composition is not known. Okay, it's more so just kind of a slew of nutrients. We might know we might not know exactly what's in there. It's typically going to have, we'll say, contain some kind of extract. Okay, and I'll just put that in parentheses. So an extract like soy extract, that's just, we've taken some soy and added some enzymes to chemically digest it to release all the nutrients. Okay, beef extract, just again, it's just digested beef. We don't know exactly what the nutrients are in there, what amounts, just a whole bunch of carbohydrates and vitamins and minerals. Okay, so anything that's complex is going to have some kind of extract in it. All right, so here's a great example of a complex medium that we use all the time in lab. It's called nutrient agar. So looking at, you know, why do we put some of these ingredients in here? Peptone, that's just a source of amino acids. And here we go. We got either beef extract or yeast extract. Like I said, just a slew of carbohydrates, vitamins, minerals. We don't know exactly what amounts are in there. Agar. Okay. I think we talked about agar last time. This is the solidifying agent. Okay, so that's how you know it's a solid media. It has some agar in it. Usually that's, you know, I wouldn't necessarily memorize an amount, but 1.5% is pretty standard. Sometimes we might put a little less in if we want kind of a semi-solid media, but that's pretty standard. A little salt here because we want an isotonic medium. Okay, that's for osmotic balance. We're going to review osmosis probably next week, but we don't want our bacteria cells gaining too much water or especially not losing too much water. All right, so just all those ingredients, water is added. Typically, we want the pH to be about neutral. And again, that's a common culture medium where a lot of organisms are going to grow just fine on. Okay, there's plenty of bacteria that are much more particular and may not grow on that, but that's just a great example of a complex medium. All right, some other labels that we might apply to media. Media can be selective, and that means there is some ingredients in there that inhibits some organisms. Okay, oftentimes that might be maybe an antibiotic that inhibits certain kinds of organisms from growing and only allows certain other kinds. So that is a really powerful thing. You know, if you're, say, swabbing the bottom of your shoe, wiping it on some selective medium, if you only want a certain thing to grow, and then hopefully kind of get rid of most of the other stuff that you're not really interested in, and only, you know, enriching for maybe that one kind of organism that you're interested in. I'll give you an example of that in a minute. Some medias are differential, and that means that it contains ingredients that cause some organisms to have a changed appearance. Okay, they look different. I don't like to use that different when it's differential. Everything grows. Okay, so there's nothing in there to inhibit growth, but certain kinds of growth will have a characteristic appearance. So it tells us something about the physiology of that organism. Okay, and media can be both selective and differential. It can be one or the other. It can be neither. All right, and we'll come back to the anaerobic media in just a second. All right, so here's an example of selective media. This example from your book, it looks like they took a soil sample. Because I've done that before. I've taken a soil sample, added some water, and added a thin film of it to some media. And that is very similar to what it looks like. All sorts of different things growing there. So here's some nutrient agar. It's not selective. You get all sorts of things growing. If you took that same source and instead put it on Sabarod's agar, the only difference between Sabarod's agar and nutrient agar is the pH. It's pH to be a little bit more acidic. And that acidity actually favors the growth of fungi. Now, there are some bacteria that grow best at pH of 5, 6, but when you're speaking kind of in generalities, a lot of fungi, that's going to favor their growth. So you see here, same sample, and you've got many more fungal colonies. Fungal colonies tend to be bigger and often are fuzzy. All right, so again, selected against the bacteria and favored the growth of fungi. Here's an example of a differential medium, and that's blood agar. That's something that you're going to use later on in the semester in lab. It actually has sheep's blood in it. Blood is a very rich ingredient in media that lots of bacteria would grow really well with that as a nutrient source. But bacteria have 3 characteristic ways that they break down or lyse red blood cells. Some of them will totally lyse red blood cells. It depends on the enzymes that they have. So here's total hemolysis, and you can see kind of a yellow appearance there. Just again, breaking open those red blood cells. Some bacteria can only partially lyse red blood cells. And so often it see a little greenish appearance there. That's because the hemoglobin is not lysed. So that heme has kind of a greenish tinge to it. And then some bacteria can't lyse red blood cells at all. So you don't see any change in the media there. All right, so here the differential agent would just be red blood cells. And we can tell something about the organism. We can put it into a group. Oh, this is a beta hemolytic organism. This is an alpha hemolytic, just from its appearance. Okay, everything's growing. Yep, question? Why is the middle one green? Because when a red blood cell is lysed by these organisms, they don't lyse the hemoglobin. Okay, so that's just a little extra FYI. You don't need to memorize these examples, but it's just an example of how we have a different appearance of some organisms on certain medias. Okay, the last thing here, and I think in my mind, it seems kind of odd to throw anaerobic media in here, but that's what your book did, so we'll do it. Anaerobic media, this is media that keeps oxygen out. Okay, so there are a lot of bacteria that are actually kind of poisoned by certain forms of oxygen, so they can't grow in the presence of oxygen, and we have some different ways that we could try to keep oxygen away from organisms. One example is some media that actually chemically gets rid of oxygen. All right, so I thought I would show you this. We're going to talk about this a little bit later in the semester, and you're going to inoculate this media in lab as well. But this is, again, a kind of media that has a reducing agent in it so that it chemically gets rid of the oxygen. So with any broth, you're going to have more oxygen at the top and less at the bottom. But some does diffuse, underneath the cap and down into that media. With this media, essentially below, a certain zone, there's going to be no oxygen. So if we want to see what an organism's oxygen requirement is, we might inoculate that tube and then look to see where the organism grows. So for example, if it's only growing at the top, that's an organism that has to have oxygen in order to metabolize. Organisms that only grow at the bottom, we would probably call an anaerobe. All right, so with those labels that we've talked about, here's the kind of question that you might see for me on an exam. And I kind of debated how to ask this. I thought, oh, I should be able to maybe just give you the recipe, but I've actually been a little bit more helpful and explained what you should understand from that recipe. So let me give you a chance to read that. Compare with your neighbor, which you would say, I'll give you about 30 seconds or a minute here. All right, I like the discussions that I'm hearing, like arguments why it's one thing or why it's another thing. So let's pick this apart. So what I told, this is a real medium mannitol salt agar. So the first thing I told you is that organisms that can ferment mannitol, which is a sugar, will turn yellow. So the organisms that can't ferment mannitol won't turn yellow. Okay, so you know that it's differential. Okay, you got a different appearance. But I've also said that only organisms that can tolerate high salt will grow. So that means if they can't tolerate that high salt, they're inhibited. All right, so they're also going to be selective. All right, so right here, the sodium chloride, 75 grams, that's actually 7.5%. So that's pretty high salt. Physiological salt is maybe closer to 1 or a little bit less, 1%. Okay, and I also, if you look at the recipe here, beef extract is one of the ingredients there. So you know that it's complex. So the correct answer here is going to be D. It's selective and differential and also complex. All right, is it a solid or liquid media? Agar is one of the ingredients. So that's going to be solid. All right, so good. Questions there? Okay. Good. So mostly I want you to know the definition there and be able to apply it, not worry about the example. All right, so as we wrap up talking about our introduction to talking about growing bacteria, I don't want to give you the impression that we are really, really good at this. There are so many organisms that we know are out there that we are actually not able to culture in the lab. Okay, like I said, if I streak the bottom of my shoe on some nutrient agar, only a fraction of the organisms that are actually on that shoe would grow on any kind of media that we put it on. So there are definitely organisms that we know are out there that we just don't have a way to culture in the lab. For instance, leprosy, the bacteria that causes leprosy. The only way we've found to grow that and study it is by growing it up in armadillos. Okay, that must have taken a lot of trial and error. The organism, the bacterium that causes syphilis, the only way we have to grow that up is in rabbits. We have not, despite lots and lots of trying, we have not found some media that that organism will grow on. Okay, so we estimate that of all the organisms, the bacteria that are in our body, which you would think we would know a lot about, we probably are only able to culture a very small number of those organisms that are actually in our body. And we have other ways to detect that they're there, DNA sampling, for example, but that's definitely the challenge of studying the microbiome, because once we get these organisms outside the body, we can't always put them in the right conditions to get them to grow and figure out if they're helping us or harming us.
So when they culture in the animal, does the animal then get the disease and die?
Yeah, so when we culture the organism in the animal, they get ill. Or well, you know, I don't even actually know. It may be that we can grow them in an animal model and it doesn't kill them, perhaps. I should look into that. I don't know enough about that, whether, yeah, we have to keep, you know, going through a lot of animals to do that. Yeah, that's a good question. It probably depends on the infection for sure. If we are trying to grow viruses, remember, there's not any media in the lab that a virus will grow in. Viruses need live cells to grow in. So we could take some nutrient agar and place some bacteria on there and then add some viruses that would grow in that particular bacteria, but they're not going to grow in the medium. Most often when we are trying to work with viruses, we grow them in cell culture, and that looks something like this. So we grow up a monolayer of cells that are bathed in this some kind of nutrient-rich medium, and then we add our viruses to those cells. It's difficult because you have to keep that media then from being contaminated by bacteria. So you often have to add antibiotics in there and work under a hood and be extra careful about your aseptic technique. So it's a whole another layer of difficulty, really. Okay, all right. So I did split that chapter 6. We just talked about a little bit of chapter 6 that talked about culturing. So there is a short assignment for chapter 6 that you can be working on. I think the rest of chapter 6 we probably won't get to until the next exam. So I have split that chapter up. We'll move on to chapter 4, microscopy and taxonomy. And it's kind of odd. Those are two very different things that they put into one chapter, but we'll go with it. So we're gonna think first about some principles of microscopy. And that's probably some information that you're gonna get in lab as well, but it's kind of important information. So if there's a little overlap, that's probably a good thing. We're gonna talk about different kinds of microscopy. In lab, you're only gonna use a bright field microscope. There's a lot of other different kinds of scopes out there that we'll talk about. I decided to not talk about staining. I'm going to leave, if you are in lab, talk about that in lab, and I'll show you some pictures, but there is some good information in your textbook that talks about some background of why we stain and different staining procedures. If you're interested, that might help you in lab. And then a little perhaps drier material, talk a little bit about taxonomy. It's no less important though. We have to have kind of a framework to work with these different organisms. We mentioned Carl Linnaeus last time as being our father of taxonomy, and we'll get into some more of his contributions. And then another name, Carl Woese. He made some really important contributions to how we think about organizing our organisms as well. So, all right, so before we talk about microscopy, a word on units. This is a kind of a nice graphic that your book provides. We're going to use some units that probably you don't use day to day. All right, so ideally, a unit that we would use to measure something would be a little bit smaller than whatever we're trying to measure. Okay, so to use, say, 1 meter as a unit to measure bacteria is not really going to be all that helpful. It's going to be kind of cumbersome. All right, so thinking about the kind of units that we need and kind of how we can see something that size, if we move our decimal place 3 to the left, that's so 0.001 meter, that's the same thing as a millimeter. Right, so you're familiar with millimeters. You can see that on a ruler. That's able to be seen with the naked eye. Okay, so unaided human eye, we can see a little bit smaller than a millimeter probably. So what sort of things are that size? Maybe the largest protozoan would be maybe a little bit smaller than a millimeter. Okay. So we want to get a little bit smaller than that. So we're going to go 3 more places to the left. So 0.00001 meters. That's a very useful unit called a micrometer. Okay, that little Greek mu stands for a millionth. Okay, so a millionth of a meter. We could also write this 1 × 10⁻⁶ meter. The micrometer is a really useful unit because most bacteria cells, their diameter, one side of the cell to the other, is about a micron. It's kind of a useful gauge or kind of an average. Okay, so typical bacteria and archaea, our eukaryotic cells are going to be a little bit bigger than that. And then, some of the organelles in there somewhere in between. If we want to measure viruses or maybe structures within bacteria cells, it will be useful to go 3 more places to the left, a nanometer. Okay, so nano means a billionth of a meter. So again, this is 1 × 10⁹ meter. There's a couple of homework questions that ask you to convert between those units. It's really, I'm not going to take the time to go through that in class, but it's just a matter of kind of knowing which way to move the decimal place to convert between them, and knowing that nano is the smallest, and then micro and milli and meter. Okay, so a nanometer would be helpful, probably more helpful to measure viruses and, again, some of the structures that are within bacteria cells. What kind of microscopes do we need? A compound light microscope is we are going to be able to see bacteria with that. We really can't make out any structures within bacteria cells with light microscopy. We're probably going to need some kind of electron microscopy to see structures. Don't worry about this scanning tunneling or atomic force. We're not going to, those are a newer kind of microscopy, and we're not going to cover those in this class. Okay. So let's look through some different kinds of light microscopy as well as electron microscopy, and think about what kind of things we need to satisfy in order to see microbes successfully. All right, so 3 general principles. The first thing that, of course, you need is good magnification, because everything we're trying to see is too small to be seen with the naked eye. So apparent increase in size. Okay, and to figure out the magnification, you would take the magnification of the ocular lenses and multiply that by whatever objective lens that you're looking through. With light microscopy, usually, we'll say light microscopy, we can get up to about 1,000 times magnification. Okay, those scopes that you're going to be using in lab, we can get up to 1,000 times magnification. We actually could make lenses to give us better magnification than that, but it actually wouldn't do us any good, because above that magnification, we start having problems with the physical properties of light inhibiting how clearly we see something. Okay, we start to get impacted by limits to resolution. Okay, so what is resolution? We're going to define that as the ability to distinguish 2 objects as separate. Okay, under the microscope, or whether you're looking at a telescope or something like that. Okay, so that's a term that's a little bit harder to wrap your mind around. If I put 2 dots on the board here, from where I'm standing, I can actually tell that there's 2 dots, that there's a space in between. I imagine maybe the back of the room that just looks like a single dot. You can't resolve that they're actually separate structures. So that's kind of what we're getting at here. Good resolution is when that space between 2 objects is really, really small and we can still tell that they're separate. For instance, with light microscopy, the cell wall and the cell membrane, our resolution is not good enough. They just look like one structure. We can't resolve that there's space between them and they're actually 2 separate structures. So don't confuse this with being in focus. We always assume that our scopes are in focus. This is more of a property of a microscope. If we think about how we can get the best resolution that we can, let's think about what determines the resolution. The resolution is equal to the wavelength of light divided by the numerical aperture. Okay. And the numerical aperture, we're going to define that as the range of angles of light. Accepted into the lens. Okay, well, we'll pick that apart in a minute, so we'll come back to that. First of all, understand that the resolution or the resolving power, okay, this is really, it's a measure, it's a distance. Okay, so to have good resolution, we want a small number. We want this to be a small distance. Okay, so then if we want this fraction over here to get smaller, either, so to improve resolution, either we want to shorten the wavelength of light, or we want to increase the numerical aperture. Okay, so think that through, like throw some numbers in here. So if we have the fraction 2/4, okay, if the numerator gets smaller, that's going to be a smaller distance. Okay, 1/4 is smaller than 2/4. Okay, 2/4. So numerator gets smaller, or the denominator gets bigger. Okay, so in other words, 1/8 is a smaller number than 1/4. Okay, so how do we do that? How do we shorten the wavelength of light? Well, I'll come back to this page in a minute, but if you look at the spectrum of light, okay, remember visible light is somewhere between 400 and 700 nanometers. Okay, so that means when you measure the crest of one wavelength to the crest of another, that would be somewhere between 400 and 700, or the trough from one to the trough of another. Okay, so if those wavelengths are larger, we get things like infrared lights and radio waves and microwaves. As those wavelengths get smaller and smaller, we get things like UV lights, X-rays, gamma rays, and electrons. Oh, if we use electrons as our light source, okay, even smaller wavelength, that helps explain why we get such amazing resolution from electron microscopes. Okay, so we could perhaps, and I've never tried this in lab, what if we used a lamp with purple light rather than white light? Maybe we would get a little bit better resolution. My guess is that wouldn't make a huge difference, but in theory, it would. Okay. Let's think about how we can change this numerical aperture. So in lab, we are almost always going to use an objective lens that allows us to use oil. We almost never even bother to look at bacteria without using an oil immersion lens. So the reason is because that improves resolution. So here's a lens that's not designed to be used with immersion oil. So typically the low power, the scanning power, maybe the 4x objective is not designed to be used in oil. So when we have the light source down here under the stage and the light comes up through, say here's our bacteria cells, our specimen, we don't typically use a cover slip in lab, but whatever, it's pictured here and that's fine. When the light comes up through the glass and hits the air has a different refractive index than the glass. So that typically causes the light to bend off in many different directions. Some of the light will go up through into the lens, but a lot of it is kind of lost. Okay, if, however, we use an oil immersion lens, so in lab, the 100 power lens that you'll use quite a bit is designed to actually go down into a drop of oil. So you find, here's your cells. Again, we're not typically using a cover slip. You put that drop of oil, usually right down there, right on your cells. And then if you're in focus, that oil immersion lens will go right down in that oil. So now when your light comes up through there, the refractive index of the oil is the same as the glass. So you get much less bending of the light. more of it comes up through the lens. Okay, so I tried to just write that out here. This actually increases the numerical aperture because we have a larger angle, larger range of angles of light that come up actually into the objective lens. You very quickly get into a lot of physics of light when you're talking about microscopy, but that's the basis. Okay, so when we use oil in an oil immersion lens, okay, that's going to increase the numerical aperture, which in theory should give us better resolution. It's going to make this fraction a smaller distance. All right, questions so far? All right, so we need good magnification. Hopefully we have good resolution. The other thing that we need in order to see ourselves well is contrast. Okay, and contrast is the ability of cells to stand out from the background. If your cells look the same as the background, even if you've got great magnification, great resolution, if you can't find them, then it doesn't do you a lot of good. Okay, so most of the time, to improve contrast, most of the time we are going to stain our cells. Okay, so here's an example. I like this figure from your book here. Here's some cheek cells. I don't know if they still do this in Bio 120, but back years ago when I used to teach that, you would take a swab of cells from your cheek and rub it in a drop of water and see if you could find those cells under the microscope. If you don't add any dye to it, it looks something like this first picture. Not impossible to find, but pretty difficult. You have to be pretty savvy with the microscope to be able to see those. So normally you add a drop of something like methylene blue, and then it's quite easy to see the cells. They have good contrast, and you can find them. Incidentally, what are these little dots here? Any guesses? Okay, those are bacteria. So you kind of get an idea of how small bacteria are compared to a typical eukaryotic cell. All right, so one of the drawbacks with staining cells is that it does kill the cells. Right, every time, always going to kill them. You're not going to see cells swimming around after you stain them. Part of the staining process involves heating the cells so they kind of will stick to the slide, and the chemicals that we use in staining are going to kill them. So you can see them, but unfortunately they're dead. Okay. If you want to improve contrast without staining, really the only thing that you can do that will help a little bit is to dim the light. Okay, so there are different ways to adjust the lights on the microscope. I would encourage you, if you're in lab, to be messing around with that all the time to try to see the cells as best you can. If you dim the light a little bit, then kind of the edges do show up a little bit better. And again, it takes some practice, but you can usually see cells a little bit. But usually we're just going to stain them. Okay, now there are different kinds of microscopy that allows us to see cells when they're still living. All right, so let's talk about some advantages. These are the types of microscopy that I want to at least discuss very briefly. I just thought I'd throw that list there. And so you can see most of them are using some kind of light, but then we'll also talk about some advantages of using electrons as well. All right, so light microscopy, this is the most common kind of microscopy. It's a lot cheaper, it's a lot easier to prepare your cells. Most often you will see bright field microscopy. Okay, so that's the kind of microscopes that we have in lab, the only kind of scope that we'll be looking through in lab. This figure looks pretty close to what the scopes that we have in lab. I'm not going to go through the parts of the scope. There is a homework question where you have to label the parts of the scope. It's reasonable that you're familiar with that, but I'm not going to test you about it here. Your lab professor might want you to know all those parts. We are using compound scopes. So that just means that there are a series of lenses in that microscope rather than just one lens. Okay, so bright field microscopy. Let's see, like I said, we get up to 1,000 times magnification. And typically the resolution is going to be around 0.2 micrometers. All right, so any structures that are closer than 0.2 micrometers, they're just going to look like the same thing. Okay. Let's see. So here is just commonly what your cells may look like when you're looking through a bright field microscope. If you stain them, if your cells are pigmented in some way, then you don't have to stain them, and you would get good contrast without staining them. But it's mostly like photosynthetic bacteria cells that are going to be pigmented. So most of the ones that are, say, causing disease are not going to be pigmented. These cells have been gram stained, which we'll talk more about what that is later. Okay, so a light background, typically the cells are going to be stained. Okay, there are a couple options for you to be able to see your cells under a kind of light microscopy when we don't have to stain them. Okay, so one is a dark field microscope, and this, when you look at this microscope, it looks almost the same as a light field microscope or a bright field. You might not be able to tell the difference just by looking at it. What's different is that it has some kind of a stop or an obstruction down near where the light source is. So basically, I'm not really sure why they put it this way, but there's some kind of stop here so that the light can only come in to the cells at a very kind of obtuse angle. Another way to draw that or to show that is this picture from an old edition of your book. So here's the picture of the stop. It's actually turned down, like they have it facing up. It would be turned down flat. And then, so the light is only going to be able to come through like the edges here. All right, so again, it's hard for me to visualize like, oh, all the physics of light here. But here, because of that dark field stop, so the cells are going to appear light and the background dark. Okay, so no staining necessary. When we manipulate light like that, it just kind of brings out the contrast and we don't need to stain. Okay. All right, so because there's no staining necessary, we can view living cells. We can watch them squirming around. And then another kind of microscopy that is really good for getting good contrasts are phase microscopes. Okay? And the new edition of your book kind of left out the explanation of what's really special about these. How do we manipulate the light? The main idea is these phase scopes. Again, it looks very similar to a bright field microscope, so I didn't show you a picture of that. But when light rays are in phase, they're brighter. And if you have some rays that are out of phase, so here we've got a trough matching up with a crest matching up with a trough, then that's going to give contrast when the light rays are out of phase. Okay, so contrast is created because the light waves are out of phase. So basically within this scope, here's our bacteria, which is going to, the light rays that go through the bacteria are going to set off, you know, be pushed back maybe a quarter phase. And then there's a phase plate in there that pushes those light rays off phase like another quarter. So that deviation gives the light rays that went through the bacterium a little bit of contrast there. So that's the basics. Again, no staining needed. In order to get contrast, we play with the light there so that it gives us good contrast anyway. All right, so an example of what these different kinds of sorry about that. Lost my, there it is. Okay, so here's a nice graphic from your book that compares these 4 different kinds of light microscopy. All right, so here, the first one, the bright field, when you don't stain the cells, the contrast is not very good. Okay, LM stands for light microscopy. If we use a dark field microscope, like I said, black background, the cell or the structure will look brighter than the background. Phase contrast, again, because of that phase plate in there, we get good contrast without staining. We often see a little bit of a halo effect here. So that's just kind of an artifact of playing with those light rays. A different kind of phase contrast microscopy is called Normarski. Again, this is a kind of phase contrast. And you can see that image is a little bit cleaned up. You don't see that halo effect there. It almost have a nice, almost 3D kind of look to it. That is accomplished because the Normarski scope uses gradients of light somehow to accomplish that. And again, that gets kind of beyond my capacity to kind of figure that out in my mind. But again, gradients of light, and you don't need to stain. Okay, so if we use light that is a little shorter in wavelength, okay, that's going to be the good thing about using fluorescent microscopes. Okay, they use UV lights. So remember, shorter wavelength, and that's going to improve resolution. Okay, and fluorescent microscopes, of course, you don't want to look into ultraviolet light. That's really bad for your eyes. So they have a kind of a plate in there to shield your eyes. So you don't have to worry about that. But this uses fluorescent dyes. So we use special kinds of stains that will fluoresce when they're hit with ultraviolet light. The beauty of this can be a really nice diagnostic tool. This graphic here, this is a sputum sample. So sputum is not just what you spit out, but it's what a patient would really kind of hawk up from their lungs a little bit. So if you have a patient who's suspected to have tuberculosis, you might take a sputum sample, look at that. If you looked at that under light microscopy, it might look like something like this, you know, just kind of some junk in there. But if you add a special fluorescent dye that will adhere only to a constituent of the cell wall of that bacterium that causes tuberculosis, then when you look at it under a fluorescent microscope, if those bacteria are there, then only they fluoresce. Okay, so here's a patient that has an active case of TB, tuberculosis, and again, you could diagnose that just by doing some acid-fast staining. Additionally, this is going to be an even more powerful tool because we can tag antibodies to fluorescent dyes. And we're going to talk a lot more about antibodies later on in the course, but we can make antibodies that are going to recognize almost anything. So for instance, we can make antibodies that are only going to recognize a certain protein on the cell wall of Yersinia pestis. Yersinia pestis is the bacterium that causes bubonic plague, right? And you could tag a fluorescent dye to that, add that antibody and fluorescent dye to your sample. And if it fluoresces, well, then you know that it's that Yersinia pestis. Okay, so again, really kind of exciting diagnostic tool. There's a another kind of microscopy that also uses a form of UV light. Confocal microscopes actually use ultraviolet lasers. Okay, and fluorescent dyes. So confocal microscopes are often talked about as being kind of a nice bridge between light microscopy and electron microscopy. They use light, ultraviolet light, but they can kind of, we get a little bit such better resolution with that, that's almost as good as some of the electron microscopes. So for years, we were thinking about getting an electron microscope here at Grand Valley. It's very difficult to prepare specimens for electron microscopy. So we ended up getting a confocal scope instead. We have one over at Kinshie. And when you look at your cells, it can look, this is an example of a confocal micrograph. These are some brain, cow medulla cells. You actually, the computer takes sections of the specimen and then puts it together on the screen. You view it through the screen. And actually, you can make it look 3D. So this image that you see, you can actually turn it to the side or turn it from this angle or turn it to that angle on the screen in order to get a better view of that. So pretty cool instrument. Not something that you would have in your dorm room. We're getting to be a little bit more expensive here. Okay. Okay, I'll say computer generated image. Okay. All right, so here's an example, some more examples. I don't think I posted this, but if you compare fluorescent microscopy, so the top row here is what that specimen would look at just with regular fluorescence, and then the bottom here is comparing it with confocal. You can see we just don't have quite as good resolution with the fluorescence. So the first one here is a human medulla section. The second row here, this is muscle, rabbit muscle. So just again, a little better clarity resolution here with the confocal, and the last one is pollen grain. So okay, so that brings us to the best resolution. I've tried to been kind of going along in order of better resolution as we go. Electron microscopy. Okay, so like I mentioned before, the wavelength of electrons is way shorter than visible or UV light, so get much better resolution. One drawback is, besides the expense, the cells have to be fixed a special way and viewed in a vacuum. Oops. 2U's, 1A. All right, so the problem is electrons, when they travel through the air scatters the electrons. All right, so we have to do all this in the absence of air in a vacuum. So that makes it a little bit more complicated to see our specimens. What kind of magnification do we get? Somewhere typically between 10,000 and 100,000 times magnification. So 10 times to 100 times better magnification than light microscopes. You actually can get up to a million times magnification. There's a homework question that asks you about that, but it's not commonly done. More commonly, we're going to magnify 10,000 to 100,000 times. Okay. So this is a picture of what a transmission electron microscope looks like. They're big structures. Again, you got to view things under a vacuum. You're only going to see them on a computer screen. So if we compare the difference between a scanning electron microscope and a transmission electron microscope, I'll give you some examples of what the images look like here in just a second. A scanning electron microscope, basically we get a 3D image of the surface of whatever the specimen is. Okay, so the specimens are usually going to be coated in some kind of heavy metal. Okay, so it's kind of a laborious process, not as much so as a transmission, but you've got to have a way to coat them in some kind of like gold or platinum. You put it on like a screen. The hell has to be done in a vacuum. And then the electrons bounce off that heavy metal. So I'll go ahead and give you some examples of what those images look like. The color all has to be added later. Okay, so we don't get different colors what we see on the screen, but we can add that in later. So this is a marine diatom. Here's a fungal spore. We got some protozoa here, and here's some bacteria. So it's kind of cool. You can usually, if you know what you're looking at, tell what you're looking at. Pretty interesting images. Okay, and then with the transmission electron micrograph, the electrons are going to pass through the specimen. All right, so transmit, pass through. So that means we need really thin sections of our specimen. Okay, so the specimen has to be sliced thin with something called an ultra microtome. Okay, so you all seen like at the deli, at Meijer, you've got the machines that slice the salami or whatever. An ultra microtome could slice a red blood cell in 100 sections. Okay, so that's a pretty thin blade. I don't know, I've never seen one of those in work, but the specimen preparation is really extensive. They have to coat the specimen in plastic and then, slice it up really thin. Okay, so what we see is a cross section of the specimens. So sometimes there's some artifacts from all that specimen preparation. Sometimes it's kind of hard to know what you're looking at exactly. Okay, but this gives us the highest resolution. Okay, so we can do the highest magnification and it gives us the highest resolution. All right, so here's some examples of a transmission electron micrograph. There's, different examples throughout the book, but both of these are trying to show a cell with its capsule. We'll talk about that structure next week. Again, the colors are added artificially. There's not any kind of staining or anything done there. Of course, TEM stands for transmission electron micrograph. And both of these are cross sections. So this does not look 3D at all. This is a cross section of a cell. You kind of gotta, again, have some training in what you're actually looking at sometimes. All right, any questions about any kinds of microscopy, anything like that? Okay. All right, let's move on Okay, so also within chapter 4, we have a big section on classification and taxonomy. That is something that is continually evolving as we get more information about organisms. But when we think about what is taxonomy, it's really classification, nomenclature, and identification all put together. So classification is, you could say, putting an organism into some kind of a group. All right, so, oh, we would classify this organism as a protozoa. We would classify this one as a fungi. And then nomenclature, that refers to naming rules. All right, we do have rules for naming. So when something new is described, we try to follow those rules in naming it. And identification just means figuring out what group an organism belongs to. So again, all these things, they're constantly changing as we learn new information, but we base them on characteristics of the cells, of the organisms. And again, kind of the emphasis we place on certain kinds of characteristics is constantly changing. So not everybody is always going to agree on all these things. All right, so we talked about Linnaeus last time. We mentioned him. So some of his contributions, he classified organisms based on shared characteristics. Okay, and we still do that to a big extent today. Today we rely more on things that you can't actually see in some cases. But based on the work of Linnaeus, remember this was back 1700s, you know, he started out, he said, okay, there's two groups, there's plants and animals. Okay, so we've been revising that ever since. I know that when I was in high school biology, we learned the taxa of kingdom, phylum, class, order, family, and then genus and species. Okay, I learned a mnemonic. King Philip comes over for grape soda. Okay, that's the only reason that I can remember all those. Okay, so we still use those to some extent today. There is actually another layer up there that I'm going to come back to. But when we refer to organisms, we, especially bacteria, we call them by their genus and species name. Okay, and that's Linnaeus that came up with that. He grouped organisms that interbreed into species. Okay, so if they interbreed, they're in the same species. Now, that doesn't work very well for bacteria because they don't breed that way. They just have one cell that becomes two. So now we say roughly for bacteria, species typically are going to share about 70% of the same DNA. We use a lot of other characteristics like their shape and some of the things that we're going to talk about soon. But typically, that's what we do for species. Linnaeus was also famous for advocating the use of binomial nomenclature. Okay, that means every organism is going to have two names. Okay, so organisms are referred to both by their genus and their species. So Escherichia coli. Okay, Escherichia is the genus, coli is the species. And when we handwrite that, we're going to do that grammatically correct. We capitalize the genus and we lowercase the species and we underline genus and species. Okay, if we're typing it out, we're going to italicize. Okay, you are microbiology students and you need to get in the habit of doing that correctly. All the time I cringe when I see, you know, a bacteria name or whatever in the newspaper, and I almost never do that correctly. So capitalized genus, lowercase species. Now, the genus actually, Linnaeus said the genus is supposed to be a noun and the species should be an adjective. Theodore Escherich, for example, he actually named this organism Bacterium coli. So, I mean, coli means of the colon. So that was an adjective, bacterium, noun. Later it was renamed in his honor. So a lot of bacteria are named after people. That's no surprise. Linnaeus said we're going to give every organism Latin names. Okay, so if he was Swedish, what if he said all the organisms have to have a Swedish name? People might not have gone along with that. So we try to make the names sound Latin by adding, you know, an AE or an I at the end. Okay, a lot of times people ask me, when can I abbreviate the genus? Okay, more often we're going to call this organism E. coli. You are supposed to, the first time you mention an organism, write out the whole genus and species. And then maybe later on in the paragraph, if you refer to that same organism, then you can abbreviate the genus. Okay, so that's the rules for abbreviation. E. coli is just kind of so common that we often just refer to that. Okay, so why was it important to have all this classification, these naming rules? This is a graph that, a table that I didn't post because I didn't have a very good, I don't remember exactly where I got it. Actually, I got it from an encyclopedia over at the library. This is old, but it didn't have a very good citation to it. But anyway, I wanted to draw your attention to this because this is just an estimation of the numbers of viruses and bacteria, fungi and algae, different species that are out there. So you can see as of 95, so that's been a while ago, we had described, say, 4,000 species of bacteria. But due to evidence that we had, we suspect that there's somewhere around 1 million different species. So we have a long ways to go. As we discover new species of viruses and bacteria, we want to have a framework in which we can kind of fit them right in. So it's important to try to classify things correctly so we understand relationships. Okay, so I know we're almost out of time here. Linnaeus said 2 kingdoms, right? He said, let's have plants and animals. So we've been revising that ever since. 1969, at that point, we had said, oh, nope, there's more. There's 5 kingdoms. So we said, so we had the plants, the animals, fungi, protozoa, and then the Monera is kind of an old-fashioned term for bacteria. Okay, so that was as of 1969. Okay, we kind of moved away from using that kingdom system anymore because today we place greater emphasis on genetic comparisons. So we look more at ribosomal RNA, at DNA when we're comparing organisms rather than just what they look like. Okay, we try to take into account phylogenetic relationships, and that is a fancy name for evolutionary relationships. Okay, and that's due to some work done by Carl Wose. And where he, I think that'll be a good place to start next time. The work of Carl Wose. So next time we'll finish up classification. We will probably get started into some structures. So remember, I will see you next Wednesday, not Monday. Little homework you can be working on. Have a good weekend.